In threshold gating, a competitive protein binding interaction modifies the response behavior of a genetic circuit. Suppose there is a promoter, shown as the blue arrow, which is controlling the expression of some gene in pink. There is also a transcription factor in blue that acts upon the promoter. In the example we'll discuss, the blue transcription factor is an activator, but the general point of threshold gating applies to either activators or repressors. So if there is some amount of the blue protein present, it would cause activation, and we could express the fractional activation with a Ka over 1 plus Ka binding polynomial term. Furthermore, let's consider the extreme case in which the binding is subnanomolar and thus the DNA titrates out the activator. Thus, I illustrate that activator as bound to the promoter and causing transcription initiation. Now consider that I add a second green gene encoding a protein that inhibits and sequesters the activator. Assuming that it binds very tightly and its concentration exceeds that of the activator, then it will titrate out all the activator into these non-functional complexes. Thus, there isn't much activator around to bind to the promoter and it goes unoccupied. Finally, consider that we add a little more activators such that it is more concentrated than the inhibitor, and thus the free activator can bind to the promoter and initiate transcription. Thus, as you ramp up the amount of activator, output transcription is 0 until some threshold is reached, and then the transcription jumps dramatically with a relatively small addition of activator. In this paper by Cross and co-workers, they use this threshold gating motif to construct a genetic circuit. At left here, they illustrate that the activator activates some promoter, but A can also participate in complex formation, resulting in an inactive complex. At right, the dotted line is the predicted response of their circuit without the competing sequestration reaction, and the solid line shows what happens if the inhibitor is present. Adding the inhibitor has two effects. First, it results in a high-pass filter in which weak signals are attenuated and only strong signals result in responses. Second, the switch becomes ultra-sensitive and switches from off to on under a narrow range of input values. Let's do some math. This first equation is derived from the equilibrium expression Kd equals A times B over AB complex concentration. The total amount of A total, is equal to the amount of free A plus the amount of AB complex. Similarly, total B is equal to free B plus AB complex. I won't derive it all in detail, but what you do is first solve the equation for A, then substitute in A total minus A for AB, then substitute that in for B, and finally you can solve that for A using the quadratic equation. So this is an expression for free A. In Shea-Ackers, we describe the fraction of the promoters active for transcription as KAA over 1 plus KAA. So we could substitute this expression for A into that, and we get an appropriate model for the system. That would be all correct, but it would be pretty ugly. That derivation will deal with the edge cases where the binding interactions are moderate in strength, but we're going to make a simplification to make the math look easier. Instead, the authors do some simplifying math. They model the transcription rate as some constant rate O min plus O max times A over A plus K. This looks a little different than our Shea-Ackers math, but it's very similar. If we derive our Shea-Ackers model and include a basal transcription rate for the promoter in the absence of A, and we also assume that the degradation is 0th order instead of 1st order, then we'll get this equation. They'll assume that A and B bind tightly to one another. This is equivalent to saying that B total divided by KD is much greater than 1. If this is the case, then the system can be modeled with two equations. When A total is less than B total, the transcription rate is the O min rate. When A total exceeds B total, then we use the A over A plus K term, substituting in A total minus B total for A. They experimentally test the math in yeast. They construct two genes. The CEBP alpha gene is the activator and DN is the inhibitor. They examine different promoters for each construct to scan the parameter space. If the activator is active, it causes YFP expression from a downstream reporter. Experimentally, the observed data looks quite similar to the model. Each colored line represents a different strength promoter on the inhibitor DN. For weaker promoters, the system has the slow and graded response of the inhibitor-free model. As the promoters get stronger, the more inhibitor is present, which shifts the response towards the right by soaking up activator. At the higher promoter strengths, ultrasensitivity is clearly present.